for inviting me to this meeting. I am so sorry that I cannot attend in person at this time, but I plan to attend in person next year. So I hope to see many of you next year in person. And so, as you know, I have been working on Wolfram syndrome in the past 20 years. So today I'm going to talk about therapeutic development for Wolfram syndrome. Before I start, I am very, very sorry for the loss of Queen Elizabeth. I met with her once when I was in kindergarten. I was part of the dance team, and we went to the Olympic Stadium in the center of Tokyo, Japan, and Queen Elizabeth came, and I was part of the dance team, and I had a chance to meet with her. So I'm very, very sorry. Okay. Thank you again, Tracy. Thank you again for Wolfram UK for inviting me. And these are my disclosures. My dog and my sons. And my work on Wolfram syndrome is a definitely collaboration with many scientists and family members all over the world, including Wolfram UK and Dr. Tim Barrett team at University of Birmingham and the affiliated hospitals at University of Birmingham. So as you know, Wolfram syndrome is characterized by diabetes mellitus, usually early onset, and optic nerve atrophy, deafness, neurodegeneration, and there are two causative genes, WFS1 and CISD2. And for diabetes mellitus, we have treatments such as insulin, GLP-1 receptor agonist, metformin. And the deafness, we have some treatments such as hearing aids, cochlear implants, but we don't have any treatments for optic nerve atrophy and neurodegeneration. So we are trying to develop treatments for optic nerve atrophy and neurodegeneration. So this is, in the past 20 years, we have learned that Wolfram syndrome is a spectrum of disorder. So typical Wolfram syndrome patients have two mutated copies of WFS1. As you know, we all have two copies of WFS1. We get one from the mother, one from the father. And if we have two mutated copies, we develop Wolfram syndrome. We found some patients who developed medical conditions due to having one mutated copy. These are so-called dominant Wolfram gene variants. And that's not the focus of my talk today, but we are also interested in these populations. So Wolfram syndrome is a prototype endoplasmic reticulum disorder. And because diabetes mellitus is the first manifestation, usually, many patients are diagnosed as type 1 diabetes, but they are usually autoantibody negative, so they don't really have type 1 diabetes. So it's important to actually set up genetic testing for patients who are antibody negative so that we can catch Wolfram syndrome earlier. And in the past 20 years, we have been working on, we have been trying to reveal the molecular mechanisms of Wolfram syndrome. And so the basically loss of function of Wolfram gene causes high levels of endoplasmic reticulum stress, mitochondrial dysfunction, low levels of ER calcium in the ER and in the endoplasmic reticulum, high levels of cytoplasmic calcium. So this is a very these are the very simplified findings of my research laboratory. And we have been also running multiple clinical studies at Washington University Medical Center. So we are running a registry and we have medical records from more than 2,000 patients. And we have biological samples such as skin, blood, urine from more than 100 patients. And we provide genetic testing at no cost, including family members. So that include anyone in the world, as long as they have, we can send their kit by FedEx. And we are also running a longitudinal study data from more than 50 patients. This is this study has been run by Dr. Tammy Hershey and Dr. Bess Marshall at Washington University Medical Center. So if any of you or your friends or anyone you know has Wolfram, please contact us. My nurse coordinator, Stacy Hurst here. There is her email addresses and her phone number. So please either call or send her email, and we will enroll you into an appropriate study. So this is the therapeutic development pipelines and timeline for the therapeutic development. And as you know, the root cause of the Wolfram syndrome is the pathogenic variants or mutations in the WFS1 gene that can lead to their downstream consequences. So downstream consequences are dysregulated ER calcium homeostasis, endoplasmic reticulum stress, and the mitochondrial dysfunction, and leading to cell death and clinical manifestations such as optic nerve atrophy, diabetes mellitus, and neurodegeneration. So we are basically trying to target different parts of the pathogenesis of Wolfram syndrome. So initially, we tried to target endoplasmic reticulum dysfunction using different types of medications, existing medications. And so the first medication we tested was dantrolene sodium. So this is the timeline of the dantrolene clinical trial. This trial has been concluded, but we found the molecule in 2014. And 2016, we got the FDA approval. No, I'm sorry, we secured the orphan drug designation of dantrolene sodium for the treatment of Wolfram. We also got the orphan drug designation from EMA in 2016. And in 2017, the IND, investigational new drug status, we secured it. And the trial started in pediatric and adult Wolfram patients in January 2017, and 21 patients enrolled. And both Snow Foundation and Ellie White Foundation, also the Silverman family, donated money to start this trial. Later on, this trial was also funded by NIH, National Institutes of Health of the United States. So here is the outcome of the phase 1b, phase 2a clinical trial of dantrolene sodium in patients with Wolfram. Dr. Stephen Stone, he was our former fellow. Now he's a faculty member and he has his own lab. And former students, Damian Evelu, now he is a resident he did his internship in medicine last year, and now he is a resident in dermatology. But both of them did a fantastic job, and many of my collaborators worked hard on this. So these are the findings. So we found the safe dose for dantrolene sodium in children, 0.5 milligram per kilogram day to 2 milligram per kilogram per day, and adults up to 100 milligram per day. So with these doses, patients don't have side effects, and their liver functions are good. And so we did see some improved beta cell functions in a subset of patients. And the patients with the greatest increase in beta cell function tended to have improved visual acuity and less severe disease. And the most common side effects were fatigue and diarrhea. So you can find these results in this article, and this is an article open to public, so you can download PDF file through the internet. And so this study at Washington University was concluded, but the phase 3 study has been planned by a biotech company. So please stay tuned. We are also trying to target mutant WFSM protein, ER stress, and mitochondrial dysfunction with a new medication called AMX0035. So this is a medication developed by a company called Amylyx in Cambridge, Massachusetts. So this is the mixture of sonul phenylbutyrate and taurosodiol. And both of these molecules have been shown to reduce endoplasmic reticulum stress and prevents mitochondrial dysfunction. So AMX0035 has two components, sodium phenylbutyrate and taurosodiol. So this drug was originally developed for the treatment of ALS. And so the medication has not been approved for the treatment of ALS yet by US FDA, but they got their pretty encouraging results for ALS, and I hope their drug will be approved for ALS, but we are also developing this medication for the treatment of Wolfram. So we completed the preclinical study at Washington University School of Medicine, and mainly done by Dr. Rie Asada in my lab. So I will show you some results using cells from our patients. So we created neural progenitor cells using induced pluripotent stem cells derived from some of our Wolfram patients. And the neural progenitor cells are brain-like cells. And we treated these cells with AMX0035. So y-axis shows their cell death. So relative caspase 3-7 activity means cell death. And so if the number is high, that means cells are dying. So we treated these cells with AMX, then vehicle, this is their cell death levels. And the cell death levels went down by treating these cells with AMX0035. So in 3 different brain-like cells derived from 3 different patients. And these are consistent results. And we also treated these cells with PBA only or TUDCA only. And so both PBA and TUDCA could suppress cell death too. But if we mix them, it's even better. So this is very encouraging. So we started this collaboration with Amylyx 4 years ago. And these results were just published in Journal of Clinical Investigation Insight. So if you go to Journal of Clinical Investigation Insight site, you can download the data, entire data. We also looked at their mitochondrial functions in Wolfram cells treated with AMX. So mitochondrial function, there is a machine that can measure mitochondrial functions. And basically we look at oxygen consumption in these cells and oxygen consumption rate in these cells. So higher is better. So y-axis shows their oxygen consumption rate and x-axis showed their time. So we treated cells from brain-like cells derived from Wolfram patients, 3 different Wolfram patients with AMX. And so we could improve mitochondrial functions in all 3 lines. So these are pretty encouraging results. We have a technology. Then what we did was we created insulin-producing pancreatic beta cells using Wolfram patients' iPSCs, induced pluripotent stem cells. So this is a collaboration with Dr. Jeff Millman's lab at Washington University. So they have the cutting-edge technology to make a high quality insulin producing cells that can be potentially utilized for the treatment of type 1 diabetes. So they are world experts, and so we created high quality insulin producing cells using lines from patients. So GSIS means glucose stimulated insulin secretion. And AN1.1 is the line beta cells created from the healthy individual iPS-induced pluripotent stem cells. So as you can see, y-axis is insulin production, and with or without high levels of glucose. And so then W024, W121, these are the lines from Wolfram patients. So insulin production levels were much lower in insulin producing cells derived from Wolfram patients' iPSCs as compared to control cells. So we tested multiple control cells and Wolfram lines. So then we treated these cells with AMX, AMX0035. And so blue, left side is the without treatment, and right side is the with treatment, pink, if you can recognize the color. So blue, left side is without AMX, right side is with AMX. So as you can see, we saw a clear increase in insulin production of insulin producing cells derived from patients treated with AMX. So these are the very encouraging results. We also have some animal data, which was sent to FDA. So US FDA granted an orphan drug designation to AMX for the treatment of Wolfram syndrome. We created the trial protocol based on the feedback from the FDA. And we also, now we have investigational new drug status. And we plan to start a trial in November or December this year at Washington University Medical Center. And so please stay tuned. We don't have any results yet, but we hope that this medication improves the quality of life of our patients. So we are also developing gene therapy and regenerative therapy for the treatment of Wolfram syndrome. So I will go through our current efforts. So our major focus right now is to correct mutant WFS1 gene variants using prime editing. Okay, before I go, okay. Okay, so using prime medicine. So this is a personalized gene therapy. So we initially we used a technology called CRISPR-Cas9 to correct mutant WFS1 gene variants. So this was already done and published. So what we did was we corrected mutations in the WFS1 gene in induced pluripotent stem cells from derived from Wolfram syndrome patients. Then we could create, different types of cell lines. And initially we created insulin producing pancreatic beta cells. So this was also already published in Science Translational Medicine in 2020. We opened this article to the public, so you should be able to download. So we try to open all of our publications to public so that people can download at no cost. So that's what we have been doing. So you can all read, maybe too technical, but you should be able to read all of our articles published from our lab. And so, okay, so we corrected, so before the gene correction by CRISPR-Cas9, insulin production was low. So left side was before the gene correction, and right side was after the gene correction. So after the gene correction, insulin producing cells derived from patients started making more insulin. So this was really encouraging. We could also see positive data in animals. So, and as you may know, the gene gene editing technology is evolving. Initially, people, used CRISPR-Cas9, which means which involves the double-stranded DNA breaks. Then people started moving on to base editing. Then now people are moving into prime editing, which is the newest technology for gene editing. And this technology was developed by Professor David Liu at Harvard University and Massachusetts Institute of Technology. He is a fantastic scientist as well as a fantastic collaborator, just like Dr. Barrett. And so he shared all the reagents, and also his staff members share all the tips to conduct prime editing. So now we are using prime editing, new gene editing technology in our Wolfram cells, and we are trying to fix neurodegeneration, optic nerve atrophy, and hearing loss. So this is actually, I couldn't find a good picture of ear, so this is my ear. So my wife took a picture and I put it there. And 3 people are actively working. So then how do we test these new medications or new gene editing technologies? So we have 2 models, induced pluripotent stem cell derived beta cells, neurons, cortical neurons, inner hair cells, as well as retinal ganglion cells in our eyes. And so we have established the very good protocol to create high quality retinal ganglion cells in the dish. So we can make retinal ganglion cells in the dish. So in Wolfram syndrome, we see retinal ganglion cell degeneration connected to optic nerve. So we established a good method to create retinal ganglion cells. We have been characterizing these retinal ganglion cells using electrophysiology, morphology, gene expression. And we see the difference between retinal ganglion cells from patients and retinal ganglion cells from control individuals, so that we can test these gene therapy. We also created 4 different types of mice carrying human Wolfram gene variants. And these animals develop Wolfram syndrome symptoms. And so these animals, actually, the symptoms of these animals were more prominent as compared to our existing Wolfram animals. So please stay tuned. We are characterizing these animals. And this is my last one. So we are also developing regenerative therapy for optic nerve atrophy using 2 pipelines. So our strategy is to stop the progression of the disease using oral medications, then try to treat damaged tissues using regenerative medicine. So we are trying to use a regenerative factor called MANF, and we are also injecting mesenchymal stem cells into eyes. So these are the ongoing projects, and mainly done by Venu Garam, who was actually trained in France, and Cécile Delato, our good friend, was one of his committee members, and he came from France to develop new therapy for optic nerve atrophy, fantastic scientist. And Eka is a research specialist. She is also from Europe. So they are working on these projects. So what is MANF? MANF is a molecule created our own body. And so the levels of this molecule is relatively low in our body, but a MANF has an interesting property because this molecule is induced by endoplasmic reticulum stress, and this molecule can confer protection against ER stress mediator cell death, and also it activates the proliferation of cells damaged by endoplasmic reticulum stress. So this molecule has homology to the neurotrophic factor. So we have been injecting viruses expressing this molecule in eyes of our Wolfram animals. So this was Dr. Morikawa, who was a fellow in my lab, did a fantastic job. So as you know, ganglion cell layer thickness is lower in Wolfram patients. So left side is control, right side is Wolfram patient. So there is a clear difference between patients and the control. And we see the same in mice. So retinal ganglion cell thickness is lower in Wolfram mice. So we injected adeno-associated virus expressing mouth into eyes of these animals. And then we looked at the retinal ganglion cell thickness, and we could increase the thickness by injecting viruses expressing mouth. So we are continuing this project, and this is also led by Venu Garam, Dr. Garam from France, and so to Billy Ann, who is a lab technician, and Shirini Bibal, who is an undergraduate researcher. Okay, so that's all for today. So these are the pipelines in my lab. So endoplasmic reticulum calcium stabilizers, which is dantrolene sodium. We completed phase 1 and phase 2, and we are moving into the phase 3 study with the biotech company. And so they are looking into European sites. So I gave the contact information of Dr. Barrett and others in Europe. And the TUDCA for PBA, this means AMX, the phase 2 trial should start in November or December this year at Washington University Medical Center. MANF is still at the preclinical stage, but we hope that we can start this one trial. And cell replacement therapy for diabetes and optic nerve atrophy, these are also at preclinical stage. And gene editing using prime editing is also at the preclinical stage, and hope that we can move these projects forward as fast as possible to help our patients. Thank you for your attention.
Thank you, Fumi. That's really, really interesting. So we have some questions. I've had some questions come in beforehand. During the talk, I did put a message saying if anyone has any questions, then please put them into the chat or the Q&A. So while some people are doing that, I'll give you the questions that I've had come in beforehand. Is AMX, the TUDCA and the 4PBA thought to be helpful in missense variants or similar of Wolfram, which result in some residual Wolframin activity, or also in those variants like nonsense that cause complete depletion of Wolframin?
Oh, that's a really technical question. So that's probably coming from a scientist. Yes. So in the article, I think in the supplemental data, and also the data submitted to FDA, we actually tested the medication in cells derived from different types of patients. So patients who have missense variants, patients who have nonsense variants, and patients who have mild Wolfram gene mutation variants. We also tested medication in the animals. So the AMX0035 worked in all different types of cells derived from patients. So it should work in different types of Wolfram patients. One thing we did not do, which we should have done, was to test the medication in cells that carry only one pathogenic variant, so-called dominant variant. We didn't do that, but we tested medication in different types of Wolfram cells derived from different types of Wolfram patients, and it worked. So that was pretty encouraging.
Wonderful. Another question is, did the dantrolene trial work? If not, can a percentage solution be found like I'm just trying to understand gene therapy to reverse prime cause, which is gene mutation.
Yes, so gene therapy, prime editing, is to correct their mutation in targeted cells. So we are targeting eye cells. Does it reverse their symptoms? We are not so sure. So in theory, it should stop the progression of the optic nerve atrophy. Does it improve? Does it reverse the phenotypes? We don't know. Our hope is that the remaining cells start working properly, then the visual acuity may improve a little bit, but that's what we hope, but we don't know. That's a really good question.
Okay. Right, next question is someone's thanking you for your continuous hard work for WS. Their question is related to the AMX study done on the Wolfram iPSC derived cells, where the Wolfram iPSC derived cells completely devoid of wolframin, as in nonsense mutations, or were they from missense-like mutations which still have some functional wolframin?
Yes, so that's also a very technical question. So it worked in both types. And in the paper, I think we mainly showed the results from results of cells carrying missense mutations in the paper. But we also tested the medication in cells completely lacking Wolfram protein due to the nonsense mutations, and it worked too. But in the paper, if you look through the paper, I think most of the results were based on the cells derived from patients who have missense mutations. They tend to have milder manifestations.
Okay,
so that's a really good question. Yes.
Okay, I've just had a comment saying that they think chat might be disabled. So if anyone has got questions, please try and put them into the Q&A. Another thank you for your impressive work. Do you think any of your treatments will eventually help in problems related to swallowing and bladder dysfunction in WS?
Yes, so that's also a very good question. So I think oral medications we are developing, such as dantrolene sodium, and actually dantrolene sodium plus something else, and also AMX 0035, because they act on the upstream molecular mechanisms of the Wolfram syndrome. This should work on different symptoms seen in Wolfram syndrome, such as including swallowing and bladder dysfunction. So oral medications targeting the upstream should should help these problems. That's what I think, that in theory. And gene therapy targeting eyes, because we only change the gene mutation in eyes, where we inject regenerative factor only in eyes, it only works for optic nerve atrophy in theory. Is it clear?
Yeah, thank you. Okay, have we got any more questions? I've had a link put in for the AMX 0035 article, which I will send out to everyone when I send the links with the recordings on the website. So thank you, Sarah, for that. Do we have any other questions from anyone? Nothing else is coming in at the moment. So what I would suggest is, if anyone has any questions.
Tracy, there is one.
There is
Tracy,
one, yes. I've just seen it come up, yes.
Yes, so
Okay.
samples could be sent from around the world. Could you please post more information with the links of the studies mentioned to be posted later? Is this presentation available too? I guess it's available, right?
Yeah, I will be editing the video and that will be posted onto the website within the next week or two. So yes, and so if there's any links or anything that yourself or perhaps Sarah can send, and then I can have those sent out to everyone as well.
So
yeah.
Samples can be sent from anywhere in the world. So we are allowed to accept samples from anywhere. And so then then last month, I was contacted by a patient's mom in a country in Africa, and it turned out we could not get the sample from that country. So it depends on the country. Most of the European countries should be okay, and Canada, US, clearly it's okay, and some countries in South America should be okay. But from some countries, because of some systems, sometimes we cannot send the kit and receive the biological sample due to some regulations
Yeah,
or
the
other more
transport
practical
biohazard or
issues.
something, I suppose.
Yeah, more practical issues too. They don't have many FedEx branches, so regulatory problems. So it depends on the country. So please just contact us and we can look into.
Wonderful. I was going to say, so would it be they contact you or someone in your team to find out? Okay, wonderful. So as I was going to say, if anyone does think of any questions after this presentation is finished, or when they re-watch it once it goes onto the website, then obviously please do email your questions to me at WSUK. So it's at admin@wolframsyndrome.co.uk, and I will get the questions forwarded onto Dr. Urano, and then we get the replies sent out to you. That's worked quite well in the past, so that's not a problem. So on that note, we don't seem to have any others. Thank you so much, Fumi, for your time and the presentation. And we look forward to seeing you in person at our conference next year.
Definitely, yes.
You will have some more information on the trial and how it's progressing. So we look forward to you coming over then. And thank you. Oh, we've just had someone come in very quickly at the last minute. Oh, someone's asking, can you take the dantrolene sodium if you're taking valproate? So taking the sodium valproate?
Yes.
Wonderful. Oh, another one. What is the most using I can't pronounce it, idebenone treatment?
That idebenone treatment, that's
Yeah.
a really good question. So idebenone has been approved for the treatment of Leber's optic neuropathy in Europe. And many patients take 900 milligrams per day. And some of our patients have been taking idebenone in the US and in South America and in Europe. And we don't have any large-scale clinical data of patients taking idebenone. In theory, idebenone is good for Wolfram too, in theory, because it targets mitochondria.
Wonderful. Can it also be used instead of dantrolene?
Instead of?
Yeah, or they're saying, sorry, can they use dantrolene instead of the other one? I can't pronounce it.
AMX?
No, the one you've just been talking about. Yeah.
So idebenone and dantrolene sodium target different organelles. So that's a really good question. So it's just case by case, case by case.
Okay. Tim had his hands up.
Thank you, Tracy, and thank you, Fumi, for a fantastic talk as always. Always really enjoy hearing you. I just wanted to follow up on the question that a patient had. Can you take dantrolene and valproate together? And I just put in a note of caution. Be really careful. Each of these drugs has a wide range of side effects, and they can interact with each other. So if you are considering this, please do discuss it with your doctor and check that there are no contraindications or any dangerous reasons why that might not be appropriate. So
thank you.
Just a major note of caution, be really careful. Discuss it with your own doctor who will know you.
I agree. Okay. I agree. Tim, thank you so much for mentioning it. Yes, I'm 100%, I 100% agree with Tim.